do a demo for the effects of disinfectants and antiseptics on microbes, and we're also going to do the same lab at the same time. It's going to be the effects of antibiotics on microbes. It's basically duplicate experiments, just exposing microbes to different things. What I've done over here is I've kind of compressed everything down into one little area. You will be inoculating your plates at the actual stations. These other two things that are over here on this side, we have an antibiotic dispenser and then our actual active ingredient disinfectant, antiseptic. Over here, these two things will be in different areas of the classroom, and I'll show you those in a bit. But I've already disinfected this area, and what I'm going to do first is I'm going to inoculate my plates. Now, you're going to be working in groups in this lab, and what you're going to do is you're going to have 6 plates to begin with. You have to label those plates. You're going to label them with your group symbol. You're also going to label what's going to be on the plate. So you'll have 3 microbes on each set. You'll have 1 plate of Staphylococcus aureus, 1 plate of Pseudomonas, and 1 plate of E. coli. And as I said, you're going to have duplicate sets of that. So you have 2 sets, 1 for each experiment. So after you've labeled them, you can disinfect your bench. You come over here, and as I said, you're going to be working as a group, but only 1 person will be at each station. So don't have multiple people over here, increasing your risk of contamination. So once you're ready to inoculate, what you're going to need is your plates that you're going to inoculate. You're going to need 1 sterile swab for each microbe. You're going to need the tubes of the microbes. Also, and you're going to need your alcohol burner lit. So make sure that you arrange everything. You don't need your back incinerator for this experiment. That's why it's to the back. So you want to scoot everything up so that you're away from the countertop, so you don't lean against the countertop. So what we need to do is we need to we're going to do basically the same steps that we did with the loop, except this time we're going to inoculate with the sterile swab. So I'm just going to get my sterile swab out. I'm going to start dealing with my test tube. Open it up. You have pinky train, so you hold that cap in your pinky, and then you flame the tube. Now, the reason why we're using a sterile swab here is because it will allow us to get a lot of microbes from this tube. And what we want to do is we want to make what's called a lawn. So I'm going to make sure and grab a bunch of microbes. That swab's going to be all wet. I flame the tube, and then I close it. Okay, now my microbes are all on the swab here. So remember, my plate is, move this out of the way, your plate is upside down in front of you. To inoculate it, you pick it up, and you just go back and forth across the surface of this plate. You're going to completely inoculate the entire surface of this plate. Now, to make sure you've inoculated the surface of this plate, all of it, turn it 90 degrees, and then do it again. You can actually see your streaks if you hold it to the light correctly, and just keep on going around in circles so that you are absolutely sure that every section of this plate has microbes on it. You also want to roll your swab as you're doing this. Okay. And you close it up. The swab can go back in to this container here. Now you're done with this plate. You will do the same thing for the other plates. 1 microbe per plate, and so your group is going to have to inoculate a total of 6 plates. 2 Staphylococcus aureus plates, 2 E. coli, and 2 Pseudomonas plates. And when you're done with that, you'll make your area safe and ready to move on to the next part. Okay. In the next area of the classroom, which will be way over on the other side of the classroom, you will have all this set up. You have actually 2 stations with 4 different disinfectants there. And you can see the setup here. I have the actual disinfectant, which in this case is going to be chloroseptic. We have some forceps. We have a little petri dish here with little paper discs in them. What you're going to do is you're going to take your petri dish, put it in front of this. You're going to come up here. You're going to grab one of these paper discs. You're going to open up your petri dish, and you're going to put that little paper disc down in the appropriate quadrant. And put the forceps back. Make sure that you don't mix up the forceps with the actual disinfectants. So you can see here I now have a little disc in this little area here. You're going to do that 4 times. Make sure you put the same disc in the same quadrant in each plate. So Staphylococcus aureus, E. coli, and Pseudomonas all in area 1 in this particular setup right here would have chloroseptic in there. We're going to be using different disinfectants in the actual area, but you'll have 4 discs, and make sure you put the same disinfectant in each quadrant so that you can compare. And so when you're done with this plate, you will have 4 paper discs on it. You're going to put this on a pink tray, which is going to be over by the sink on the other side of the classroom. You're going to put it label side down this time. And the reason why you're going to put it label side down is because if you flip it over, these little discs have a tendency to fall down, and they'll land on the lid, and your experiment will be messed up. So keep them label side down this time. Okay? And so that's what you're going to do for the disinfectant antiseptic lab. For the actual antibiotics lab, it starts off the same. You have your 3 plates of Staphylococcus aureus, E. coli, and Pseudomonas already inoculated. What you're going to do is there's going to be a different station it's going to contain this little antibiotic disc dispenser. This one's actually going to be faster. It's going to be easier to actually do it because we have a dispenser that will put all the discs on the plate. And in this case, we're using 4 different antibiotics. Now, the antibiotics actually have little abbreviations as to what they are. So you don't have to write what's in which quadrant. But whenever you do the disinfectant lab, you will have to write down what's in each quadrant. Okay? So in order to do this part with the antibiotics lab, what you need to do is you need to take your plate, put it down, label side down, in front of the antibiotic dispenser. Okay, this dispenser has a little ring on the bottom, so it'll actually fit over your plate. But first you have to remove the lid of the plate. Then what you're going to do is put the dispenser right over the top of your plate. You're going to push this down, let it come back up, okay, and then close your plate. Now you can see that there are 4 discs on this plate right here. So this should go a lot faster. You're going to have to do this for each of your microbes. So you have 3 plates. You'll have to do this 3 times. Okay, and once again, with these discs here, whenever you put these Petri dishes on the pink tray, you will put them label side down so that they don't come off. Okay? So this should actually be a fairly quick part of your experiment. The disinfectants will take a little bit longer to actually do because you have to put each disc, you have to get it out of the actual active ingredient and then put it in the appropriate quadrant. Okay, and once again, make sure you don't cross-contaminate. Anytime you take your Petri dishes anywhere, any of these stations, as I said, these 2 things, the disinfectants and the antibiotics, are going to be at different stations. You need to make sure you disinfect before you bring your plates there, disinfect after you leave that station. Okay? And remember, there's only 1 person per station. When we look at the disinfectant stations, which are going to be on the other side of the classroom, you're going to see that there are actually 2 disinfectants per station. Okay, right now I have bleach and Listerine at one station, and then hydrogen peroxide and pine sol at the other, but it may be different for your class. And so there's only going to be, even though there are 2 different disinfectants, there's only be 1 person at that particular station. And the stations are all outlined by these little white pieces of tape. So just make sure that you give the people that are working at those stations enough room. Get out of the sterile field whenever they're working in those sterile fields. Okay? And that's going to be it for this demo.